Hello everyone and welcome to the second video on glycogen metabolism and glycolysis. So in this short video we're going to look at how the body makes glycogen, so how it's synthesized, and we're also going to look at the hormonal regulation of the pathways of synthesis and degradation. So it's important to note, as we just looked at very briefly in the previous video, that synthesis is not simply the reversal of the breakdown. They are controlled by different pathways. So we'll see here that glucose is first converted to glucose 6-phosphate and then to glucose 1-phosphate, and then it's added to a nucleotide, uracil triphosphate or UTP, and this forms an activated molecule, UDP glucose. So there's three different enzymes involved here. With hexokinase, which we've encountered before, we'll add the phosphate group to the 6th position, and then the phosphoglucomutase, which we looked at in the previous video, mutates the glucose 6-phosphate by moving the phosphate group from the 6th position to the 1 position. And then we get an enzyme called UDP glucose phosphorylase, which will add the UDP to the glucose to form an active molecule. So if we have a molecule of glycogen on the right hand side and our activated UDP glucose, when these molecules are combined, we get the addition of the glucose to the glycogen molecule and the release of UDP. And this is regulated by an enzyme called glycogen synthase. So how are these synthesis and breakdown pathways of glycogen regulated by hormones? Well, the two key enzymes that are regulated are glycogen phosphorylase for the breakdown and glycogen synthase for the synthesis. These are very, very tightly regulated pathways. Again, we don't want them to be operating at the same time, as this would waste energy for the cell. So when breakdown is active, synthesis is inactive, and vice versa. When breakdown is inactive, synthesis is active. One of the hormones that controls glycogen synthesis is called insulin, and this regulates insulin promotes the synthesis in liver and muscle. You can imagine that when you are well fed, you have lots of glucose in your bloodstream, and this triggers the body to release insulin, and it takes up that excess glucose from your meal that you don't necessarily need straight away and converts it into glycogen for storage at a later date. Conversely, we have glucagon and adrenaline, which promote the glycogen breakdown in your liver and muscle respectively. So glucagon will act on your liver, whereas adrenaline will act on your muscles, and this will promote the breakdown of glycogen to produce glucose, which will then be used for energy. So what happens in the liver? What happens at a molecular level? Well, glucagon will bind to a glucagon receptor, here a 7-transmembrane G-protein coupled receptor. And if we consider that when your blood sugar is low, let's consider that the body doesn't have a lot of sugar around. So glucagon will bind to this receptor. It will activate an intracellular signaling pathway leading to accumulation of a molecule called cyclic AMP. This then activates a protein kinase, which will act on glycogen phosphorylase kinase, phosphorylating this enzyme, and this enzyme in turn will phosphorylate glycogen phosphorylase, converting the B form to the A form, which is the active form, and thus glycogen will then be broken down. So we have this cascade of signaling, activating enzymes, activating another enzyme. So you can see it's a very tightly controlled process. In the liver, what happens to the synthesis? Now previously we looked at the degradation. Well, glucagon again will bind to the same receptor, And this same increase in cyclic AMP and activation of the protein kinase A will convert any active glycogen synthase by phosphorylating it to an inactive form, so thereby switching off glycogen synthesis. So the same hormone, glucagon, will promote glycogen breakdown and inhibit glycogen synthesis at the same time. In the liver, it's slightly more complicated. So in the liver, we have the glycogen phosphorylase B, which is sort of considered the inactive form. But this inactive form itself can be slightly activated, and this is regulated by molecules of ATP and glucose 6-phosphate. So when levels of AMP are low, or ATP is high, then you don't need this energy. We don't need to break down the glycogen, so we have the inactive T form. But when AMP levels are high, and this is triggered by the use of energy when ATP is converted to AMP, then we need to break down glycogen, and thus we have this activated R form of glycogen phosphorylase B. So we have these are key allosteric modulators. Remember, we could have covered allosteric modulation when we looked at enzymes. So ATP, AMP, and glucose 6-phosphate will bind to the enzyme and modulate its activity. If we then consider the phosphorylase in the A form or the active form, then glucose itself, so if there's plenty of glucose around, that means we don't need to have the phosphorylase enzyme active, because we don't need any more glycogen breaking down. So glucose itself will actually bind to the active R form of the phosphorylase A, slightly inhibiting its activity. Muscle activity will also stimulate the release of adrenaline, and again, adrenaline will lead to the breakdown of glycogen. Exercise generates AMP, because exercise uses up ATP, leads to the accumulation of monophosphate, and thereby stimulates any phosphorylase B, which is considered the inactive form, but to a slightly active form. It's important to note that the binding of these are to allosteric sites, converting the inactive T form to an active R form. So how else does exercise stimulate glycogen breakdown? Well, muscle contractions, we know, are caused by intracellular increases in calcium levels. And this calcium can promote the activity of glycogen phosphorylase kinase. Remember, this is the enzyme that will phosphorylate glycogen phosphorylase. So when it's calcium free, the kinase, the phosphorylase kinase, is inactive. But when this binds calcium, then it becomes partly active, and in the presence of adrenaline, remember, adrenaline will lead to the phosphorylation of the enzyme, then it becomes fully active. So just muscle contractions on its own will lead to release of calcium. This will partially activate the enzyme. Then because of the exercise, we get release of adrenaline, and this will then cause accumulation of cyclic AMP, activation of pKa, which will then lead to phosphorylation of the kinase. Of course, the phosphorylated kinase is then active and will promote the activity of the glycogen phosphorylase, which will lead to the breakdown of glycogen. So I have a couple of multiple choice questions now for you. So which one of the following statements is correct for an active muscle? And if you'd like to have a go at this, I'll just give you some time to pause the video. Okay, now I'm going to reveal the answer. Of course, the answer was C. So glycogen breakdown is stimulated by adrenaline. Adrenaline will lead to increase in activity of the glycogen phosphorylase. AMP is an allosteric modulator of the glycogen phosphorylase enzyme. Remember, it will bind not to the active site but to another site called the allosteric site, and thereby promote the activity of the enzyme. And of course, as we've just discussed in the previous slide, calcium ions will bind to the glycogen phosphorylase kinase, which again will promote degradation of glycogen. So what happens when blood glucose is low? Well, we know that glucagon is secreted, insulin levels will be low, because remember, insulin is released when glucose levels are high. So glucagon will be secreted. This will act on the liver. We get activation of intracellular signaling pathways, accumulation of cyclic AMP, pKa. Glycogen breakdown is stimulated, whereas the synthesis is inhibited, and the glucose 6-phosphate will be converted to glucose. Glucose will then be released into the bloodstream, and this is then taken up by the muscles and by the brain. What happens when you're well fed? Well, in here we have high blood glucose levels, and glucose will bind to the catalytic site of glycogen phosphorylase, and this will inhibit the enzyme, so therefore glycogen breakdown is inhibited. And in this way, the glycogen phosphorylase enzyme itself acts as a glucose sensor, because the activity of this enzyme is modulated by glucose directly. Glucagon levels will be high when you're glucagon levels will be low when you're in the fed state, whereas insulin levels will be high. Remember, insulin is released after a meal, and this will promote uptake of glucose from the bloodstream to be stored as glycogen. So therefore, it promotes the activity of glycogen synthase. And again, this will be regulated by an intracellular signaling pathway, the details of which you don't need to know for this module, but more than likely will be covered in your second year. And that concludes the second video, and thank you very much for listening. And again, the same chapters of Stria 15 to 19 give you a more in-depth view of glycogen metabolism.